Welcome to the External Medicine Podcast. I am your host, Daniel Belkin. This is my co-host and brother, Mitch Belkin, and we are here with the one and only Derek Lowe. Derek received a PhD in organic chemistry from Duke University. He then completed a Humboldt Fellowship in Germany for his postdoc before working for several pharmaceutical companies. His blog, In the Pipeline, has been continuously operating since 2002. Derek, thank you for joining us today.
Thanks for having me.
Before we get started, do you have any financial disclosures?
Well, as you mentioned in the intro, I do work in the pharma industry, and I'm on my fourth company now, but I never talk about any of their products or research on the blog, and I'm not going to do that here today. So everything I say is my own opinion, and I do not speak for my employer.
Understood. So let's talk a little bit about pharmaceutical development. So in medical school, we have to take lots of coursework on pharmacology, but we don't learn an awful lot about drug discovery. Can you give us an overview of what the drug discovery process looks like?
Sure. That actually is one of the main reasons I started the blog, because when I would tell people what I did for a living, they would be like, wow, that's an interesting job. I didn't know people did that. In fact, people thought that MDs discovered drugs. So don't panic. You don't have to. But the overview is that it's a very strange industry. And I think the single biggest fact about it, and I'm going to start at the end, the single biggest fact about it is that the success rate in clinical trials is about 15% overall. And that's a generous estimate. That varies by the therapeutic area. Vaccines are higher, infectious disease is higher, CNS and oncology are much lower, but the overall rate is only about 15% success. And I don't think there's another major industry that has such a hard cutoff filter right at the end of its development process in the most important and expensive part. Pretty much everything that's weird about my industry comes down to that. But let me tell you how we get up to that point, because I have spent most of my career not running clinical trials, not at all. I am more of a basic research kind of guy. In terms of the drug pipeline, if you want to stick with the pipeline metaphor, I'm back where the water is starting to seep out of the bare rock. I mean, way back early. It doesn't get much earlier. And that early part is, well, trying to think of something where you might want to see a drug. Where's an unmet medical need? Or where is a target that we know about, some sort of physiological target, protein, say, that we think can be drugged? What could it be good for? And we come at it from either direction. We're willing to take wherever we can get. So you might say, all right, here's Alzheimer's disease, huge unmet medical need. What have people done for it? What other plausible ideas are there? Where's a niche for us to operate? Or you might say, you know what? We just discovered this interesting pathway here, or there's this new report that just showed up in, some journal that says that this kinase or this receptor might be involved in this disease. Let's see if we can make something out of that. So you start from some sort of hypothesis like that. And the next stage is you have to work up some method of getting chemical matter for it, because I'm a small molecule drug discovery guy. I started off as a strict synthetic organic chemist, became more of a medicinal chemist, and now I call myself a little bit more of a chemical biologist. We'll probably get into what all that means. But if you're looking for chemical matter, you've pretty much got to run some sort of screen, some sort of big assay that will be very high throughput, reproducible, reliable, and relevant. It will read out numbers that you think are directly applicable to that hypothesis. So you might want to be inhibiting an enzyme or stimulating a receptor. Maybe you're going to go straight to a cell assay. That's more work. But let's say you're trying to kill off gram-negative bacteria for an anti-infectious project. That could work too, or trying to kill off some particular tumor cell line. I don't recommend you try those, because you're probably going to discover things that we've already discovered before, or you will have discovered things that will kill off bacteria, but will also kill off your own cells. That's always a problem. So we try to be a little bit more targeted, but you can do it the other way around. You can do it phenotypic and say, all right, we have an assay in cells or maybe in some small organism, zebrafish, something, even all the way up to mice, where we have a good animal model of the disease. Why don't we just start throwing compounds at these organisms until we see the behavior, the phenotype that we want? That's another way to do it. And you can find stuff that way that you would never find the other way. In fact, you can discover whole new pathways. You can also waste vast amounts of your time and money. A bad phenotypic assay is the worst of both worlds. Most of the time, though, we do a targeted assay. We go after this protein and we try to find out some sort of readout, generally something involving fluorescence, because we're very good at detecting that, very good signal to noise. Nothing fluoresces normally in most of our mammalian biological systems. We're not giving off any green or red light usually. So those work well, but there are a lot of other ways to do it. So you'll set up an assay and you'll run a lot of compounds to it. And a lot is, a few 100,000 usually. You can go up in a large pharma, you can go up to low millions. If you want to use a technology called DNA encoded libraries, you can go up to hundreds of millions, low billions if you're a real buckaroo. That's a tricky technology to get to work, but it can work. And you'd think that if you screen, X million compounds past some sort of protein target, you're going to get some real hits. Not necessarily. I have seen many, many interesting targets where you screen 1 million compounds past it and nothing actionable comes out. Some of these are kind of difficult, but you will usually get some chemical matter. And then the hit to lead team takes over and they start looking at it and they say, okay, which of these compounds look ridiculous and stupid and toxic because we've been burned by them before? Toss those out. Which of these compounds do we have other things that look close to them already in the files that we didn't screen? Let's screen those. You're looking for SAR, a structure-activity relationship. And if you're ever talking to a medicinal chemist, just get them going on the topic of SAR, and you're set for at least 45 minutes. No problem. But if we can really get an SAR, if we can say, all right, we put a methyl group here and it got worse. We put a chloro group here and it got better. We made this chain longer. We made this one shorter, this part flatter, this part curved, this part fixed. And you do all those things with synthetic organic chemistry, and you send them back around through the assay in a constant feedback loop. And at that point, you can also start optimizing for not only just potency on the target, but selectivity against other things that you don't want. That's tough, because there are some things that you don't want. Let's say you're after some receptor and it has 7 subtypes and you don't want to set off all 7. Great. Set up assays for the other 6 and make sure that you're going selective. But there are a lot of other things going on in an organism that we have no idea about. And you'll only have the fun of finding those when you get all the way into an animal assay or all the way into humans. God help you. That does happen. So you're optimizing for potency, for selectivity. You'll start worrying about blood levels as you dose rodents and the like, looking for pharmacokinetics. How long does the compound last in the bloodstream? What's its peak blood level? Would this be suitable for once-a-day dosing? Or am I going to have to reformulate? Or maybe I should just try to make a compound that lasts longer. What does it get metabolized to? Are those metabolites toxic? How long do they take to excrete? And on and on. How is this thing going to be formulated? Is this even cost-effective to formulate it that way? Or is this going to have to be like injected directly into the brain? That's not so good. You're trying to optimize all these things simultaneously, which there's no guarantee that that can actually be done. There may not be a solution that overlaps in that parameter space. Most of the time, we can come up with something that does most of what we want, but not always. At that point, you're finally ready to say, we have a clinical candidate. Now you're going to have to scale it up. The process chemists are going to have to get their hands on it and look at your route that medchem guys like me make and laugh at it, throw it away, Go to something more feasible, less expensive, more reliable, generates less of a waste stream and all that. Scale it up because you're going to need a lot more compound to do tox testing. 2 weeks in 2 animal species, and it better be good. Not just to make sure you didn't kill the animals. That's first start. Make sure that behavioral tox is okay. It didn't make them act strange. Then you're going to do necropsy and look at the organs. You're going to do histopathology and really look at the organs. And if you don't hold your breath during 2-week tox testing, you have not been doing drug discovery long enough. So if you get through that, now you're just about ready to go to the FDA and say, okay, we want to submit an IND, an investigational new drug application. That's your application to go into human trials. You tell them, look, we've got this drug, it does this, we've tested it against that, here's our selectivity data, here's what happened to the animals, we think we're okay here, give us approval to go into humans. If you do that, now you start to spend the real money. Everything up until then has been a round-off error compared to what it costs to run a clinical trial. Phase 1, of course, just in normal volunteers, as normal as you can be and volunteer to take a drug that no one's ever eaten before. Phase 1, just mostly to check blood levels, make sure that's okay. I have seen things fail there. If that's okay, most of the time it is, then phase 2, where for the first time, someone with your disease is actually going to take your drug. You better hold your breath here too, because this is where most of the failures occur. Most of the time, we have picked the wrong target, or a target that doesn't do as much as we thought it did. Or the compound not only does that, but does something else we never anticipated, and that kills it. Most of the time, that's what happens. If you get through phase 2, though, then you go to phase 3 and branch out into a much wider patient population, better statistics, more real world, better chances for your drug not to work as well as in your carefully designed greenhouse of a phase 2 trial. But if you get through that, then you can go to the FDA and say, okay, here's the data, give us authorization. That is the NDA, the new drug application, and it is a mound of paperwork. It used to be a physical mound. At my first company, I actually joined a party where we waved goodbye to a tractor trailer pulling out of the loading dock, loaded with crates of paper for the FDA. That was back in the early 90s, in the old days. And at that point, you maybe can even get on the market. So that's drug development in a nutshell. So now you know how to do it, and you can go round up some venture capital and give it a try yourself.
How long does all of that take?
Years. It really varies. I think the absolute flat out pedal to the metal record was set by Pfizer just recently with Paxlovid, the protease inhibitor for the coronavirus. Pfizer had several legs up, though. They already had been doing viral protease inhibitors, so they had a lot of good compounds just sitting on the shelf ready to be tested. They threw a vast amount of resource at it, and there were a huge number of patients ready to go for a clinical trial. Enrollment was fast, and frankly, infectious disease always runs faster than most of the others anyway, because it's a very hard endpoint and very meaningful. There's no arguing about whether or not your infectious disease endpoint was meaningful or not for the most part. So I don't think we're ever going to see anything faster. I don't think we're going to see many things that are even close to that speed, a year and a half or so. Beyond that, you're looking at several years, depending on the disease. Alzheimer's trials are very large, slow to enroll, and slow to run because it's a slow-moving disease. So if you've got an Alzheimer's drug, you're looking at a nice, good, solid 10 years, I'd say.
It's interesting because, and I think it sort of speaks to why mouse models are so problematic when it comes to human diseases, because it's easy to infect an animal with a virus or a bacteria and then give it something to try to kill that. It's much harder to say this mouse has something that's exactly like Alzheimer's or schizophrenia or something like that, probably contributes.
You just picked 2 of the best examples of why that's so difficult, because we are the only animal that gets Alzheimer's. There are no other mammals or anything that we know of that truly gets Alzheimer's the way we get Alzheimer's, People have tried to engineer all kinds of rodents to do it. I don't believe any of the models. And schizophrenia, geez, I mean, all that CNS stuff, schizophrenia, depression, OCD, bipolar. How do you tell if an animal is mimicking the human course of bipolar disease? Heck, how can you tell sometimes if a human is mimicking the human course? There are so many different presentations of these diseases and syndromes in humans. It is extremely difficult. So yeah, infectious disease, the animal models are very clear. Diabetes, they're pretty darn good. We actually have some pretty realistic animal models of type 1 and type 2. They start going downhill after that. And at the very bottom are central nervous system.
It's interesting, diabetes being the model that we have something that most directly relates to humans, and that's obviously something that we have a lot of different drugs that actually targets.
Exactly. Exactly. It's no coincidence. And there are also, of course, a lot of mechanisms that you can think of for diabetes, all sorts of things, insulin secretion, insulin sensitivity, glucose transporters, also insulin degradation and half-life. You can think of a lot of stuff like that. Weird sensitizers like metformin that we don't even know how metformin, aka glucophage, we don't even know how that works yet. We really don't. There's a huge argument that's been going on in the literature for years, for decades, about its target. It's probably got something to do with AMP kinase, but it's probably got something to do with other things too. And you can start a food fight by asserting that you know what the answer is.
Jumping back to the hypothesis phase, where it seems like a lot of the intellectual work is done to identify targets, there's been a lot of emphasis now on computational methods like machine learning and AI to help aid or improve drug development and discovery. How confident are you in those sorts of methods of actually being valuable in the development of drugs for specific targets?
Right now, I am not confident at all. Truly not. But that said, I see no reason why these technologies can't work. I don't see any reason, any mathematical or physics or biology reason why you can't compute your way through some of these questions. It's just that it's a very hard problem and we don't really know how to do it yet. So there's, of course, a huge amount of work going into this because, I mean, think about that 85% failure rate. If you could go up to just falling on your nose 3 out of 4 times, that would be a huge improvement. So anything that would help predict success would just be overwhelmingly useful. But it is extremely hard. In many cases, we don't even know what we would be turning those computational tools loose on, like toxicity, for example. Most of the toxicity things that kill drugs are off-target effects that came out of the clear blue sky, or on-target effects that kind of came out of the clear blue sky either because we didn't realize that the target did that same thing. How are you going to compute this? Machine learning will only rearrange the knowledge you already have and find trends and patterns in it. What if it's knowledge that no one has yet?
Jumping back to you saying that clinical trials are the most expensive part of the process, I'm curious, other than the truckloads of paperwork that's required by the FDA, what makes clinical trials so expensive?
Yeah, it's a labor-intensive process, and it starts just with patient enrollment. Tracking down the right patients that fit the profile that you're looking for, making sure that they have the disease as you understand it, and that they don't have other conditions that are going to warp your data. You really want to do a very careful job there. It's kind of like painting a wall, where you want to spend a lot more time on the prep than you do actually applying the paint. So patient selection and enrollment is tedious and expensive. Then running a properly statistically valid trial, and I'm one of these guys who says that double-blinded randomized placebo controlled is the way to go. That's also a lot of work too, because you're going to need more patients than you would figure if you're running a design like that, because you want a nice, robust placebo group, and you may have several arms to the experiment. How about this dosage and that dosage, and this dosing schedule and that dosing schedule? Well, this rapidly gets out of control. You're also going to have to figure out how logistically to do this, because any really decent sized clinical trial is probably not going to be run at one center, one hospital, one medical center. You're going to have to coordinate this over several and make sure that everyone's doing it the same way, the right way, and that these data are handled properly, because mishandling the blinded data, of course, will blow you up immediately. Now, there are specialists who do this, and there are companies that do nothing but come in and help you run clinical trials, but they want a lot of money to do this. Then you have just the time involved, because infectious disease is pretty fast. Oncology, sadly, is pretty fast. A lot of the others are not so fast, weeks, months, even years when you get up to Alzheimer's. And you're going to be taking your clinical readouts all along the way at pre-specified intervals. So we're talking hundreds, thousands of basically short wellness checks and physician visits and blood draws and checks of reflexes and whatever else you're looking for that have to happen on time, every time, and the exact same way every time. So it really adds up after a while. You get up into hundreds of thousands of dollars per clinical trial subject for a lot of these trials by the time you're through with it.
Let's talk a little bit about the FDA, which you brought up in the regulatory process that brings drugs to first being able to even be tried in humans and then eventually to be approved for use in humans for specific indications. Depending on who you ask, either the FDA is run by Big Pharma and approves drugs much too quickly, or alternatively, it's way too slow and it prevents the development of life-saving drugs. Which side of this fence do you fall on?
Oh man, those are exactly the two poles. And there are people who manage to believe both of those simultaneously too. They must have very advanced compartmentalization in their forebrain. But yeah, those are the two sides of it. And this is why I do not envy the FDA at all. They have an extremely difficult job, because those are the two kinds of flack that they can catch. As someone who's been in the pharma industry, I can tell you that we do not run them. If we ran them, we wouldn't get any drugs rejected, now would we? Now, the thing is, you don't hear as much about a lot of the rejected drugs. Sometimes there'll be a big one that, everyone was anticipating, hotly watched Alzheimer's drug fails in phase 3. Sometimes you hear that, but a lot of this stuff happens quietly because the companies themselves don't feel like going out and blowing their horns about it. The big ones don't, and the small biotechs don't. Now, mind you, their investors know all about it, and their stock goes out and drops 85% the next morning. But a lot of clinical failures happen quietly. Success has 1,000 fathers, and failure is an orphan, as they say. I often emphasize this on the blog. I'm saying, okay, guys, here's this big trial for whatever that just failed. This has been going on for 6 years, and I estimate that this probably cost about $800 million, and all that money has just gone straight down the tubes because this compound is dead. We learned not to do that. So we got something for the 800 million, but not much of a return compared. So we do not run the FDA because our trials do fail. And I have actually had to change jobs because of a failed clinical trial, because the company got in such big trouble financially and was expecting to have something in their pipeline that suddenly wasn't there anymore. On the other hand, while I sympathize with the people who feel that the FDA is just a roadblock to progress, these range from patient advocacy groups who clearly have a very strong interest in this, all the way out to rather theoretical-minded libertarian types who are just like, people should make their own decisions. If something is proven safe, people should have the right to decide if they want to take it or not. Just run trials for safety and let the chips fall where they may. That drives me nuts too, because I think that these people have a sunnier view of human nature than I do. I think if you did that, you would see, well, hell, if I could just get a conscience-ectomy under that kind of regulatory regime with no conscience at all, I would be selling tablets of pressed dehydrated grape juice for Alzheimer's. My tablets have phytonutrients in them. They have antioxidants. I think they're going to be great for Alzheimer's patients. And they're safe too. I ran a trial. No adverse effects. Line up. They're only $1,000 a pill, and they're FDA approved. Come on down. I think you would see a ton of that stuff if all we did was test for safety and not efficacy. So again, that's kind of a reductio ad absurdum, but there are some people who are kind of ad absurdum out there and would like to see that kind of regulatory regime. In the real world, the FDA, I think, is probably hitting it about right when people are bitching at them from both sides. I think, though, that the recent approval for Biogen and Eisai's Alzheimer's antibody was the worst decision the FDA has made since I've been in the industry. That was terrible. I do not think that that antibody worked, and I do not think it should have been approved. So there's one where I think the FDA should have been more of a roadblock.
So we had Alex Tabarrok on the podcast a year and a half ago, and he has written a lot about the topic of regulatory nationalism. You know, the idea that, you know, in America, we only take drugs that are approved by the FDA. We can't take drugs that aren't approved by the FDA, that are only approved by the EMA, which is the European counterpart to the FDA, or whatever the alternative in Japan is, or whatever, all these different regulatory agencies. Why should, I guess my question is, should Americans be allowed to take drugs that are approved by the EMA or the Japanese equivalent of the FDA, or should they only be allowed to take drugs that are approved by the FDA?
Yeah, it's a good point. And yeah, I've talked with Alex about these issues a bit over the years, and that's one where he and I actually do probably agree. I think the EMA decisions are generally very solid, and they think the FDA decisions are generally very solid. I think Japan's regulatory agencies are pretty good too. Although, to be fair, Japan for quite a while was a little bit more lenient on efficacy. They would be more for safety. So there used to be more, probably still are, more, say, Alzheimer's drugs approved in Japan, even though they really didn't have much efficacy, but they wouldn't do any harm. So the FDA and the EMA, I think that interchanging regulatory decisions, I think frankly, it'd be fine with me. Now, of course, we have very different systems for paying and delivering these drugs, and that's where things might get complicated. But as far as the sheer evaluation of safety and efficacy, yeah, I think the FDA and the EMA are pretty much two sides of the same coin, or should be.
What are your thoughts on intellectual property and drug patents, and specifically with respect to how well titrated are the lengths of drug patents?
Yeah, it's true. There are some weirdnesses in that. That's why pharmaceutical IP is a whole specialty, even among lawyers. Patents are a thorny subject because there are people who are infuriated by the entire idea of a government granted monopoly, which is what a patent is. On the other hand, it's difficult to come up with a system that works as well. The only thing I can think of is awarding prizes. Let's say right now we award the prize of market exclusivity. You could also say, okay, we're going to award a different prize. We're going to award a prize of this fixed amount of cash. You know, that's what we'll give you if you actually do this. And then your compound, you know, goes instantly generic. And that could work, although I think the people who like that idea aren't quite ready for the figures that would be attached to that appropriate amount of cash step. But the problem with patents is that they do exist, and they don't last forever. A lot of people I talk to about patents outside the field completely seem to imagine that it's like copyright. I think U.S. copyright law is insane. And the Sonny Bono legislation years ago made it even more insane. Copyright in the US is way too long, and people think patents work the same way, that once you've got a patent, it's yours forever. It's not. as it is now, the patent terms mean that if you have a drug that took a very long time to develop, or maybe had some difficulties at the regulatory stage, you may only have a few years left by the time it actually gets patented. So in that case, you're going to be trying to make as much hay while that sun is shining as you possibly can. Thus, very high prices for a short time. Because when these things do go off patent, the price, of course, drops tremendously, except in biologics. And this is where we get into the antibodies and the proteins like insulin and the like. The FDA, and just in recent years, has been doing more work trying to get biological generics going. But for small molecules, the price drops off a cliff. And I also was at a company where the transition to a big compound going generic was not handled very well. And it left quite a bit of devastation in its wake. But patents mean that your drug does go away. And every single small molecule drug that's out there has a clock ticking over it with a sword about to drop on its head. I don't think a lot of people realize that. So in order to avoid that, companies try every trick they can possibly come up with to keep that sword from dropping when it's supposed to. There are various extensions. You can get an extension for running a pediatric trial. You can get extensions for all sorts of other little things. You can try to change things up just enough to get a new patent on a somewhat different version of the compound that had better have some novelty and better have some utility, because those are the patent criteria. But if you can get it patented, you can turn around and say, okay, no one should be taking that old junk. We've got the new improved version over here, which also still happens to be under patent. So people try all sorts of tricks like that, and not all of those tricks make us look good, to be frank.
Can you talk a little bit more about why biologics currently do not have the ability to have generics, making it seem as though insulin or some other compound is forever going to be owned by whoever has those patents?
Now, it's a good point, because biologics are simply a lot more complicated to manufacture and to assay than small molecules are. There are also a lot more variations you can run on them, because when people talk about insulin, they are often talking about the Banting and Best insulin that was discovered back when and won a Nobel Prize. That is not the insulin that most people are taking. Insulin has been modified over the years with a lot of changes to some of the key amino acids. It's had a lot of modifications to it, like adding polyethylene glycol chains and other things to extend its half-life, etc. So there are a lot of, I guess, neo-insulins out there, which is what the majority of people in the developed world are taking. And those are expensive because they were developed as new inventions, and they are actually superior to plain old insulin. So the problem is that when you start talking about making a large protein, which is what most of these biologics are, insulin being one of the smaller ones, when you go out to antibodies and things like that, there are so many weird variables in the manufacturing, because you're going to be making these things generally in some sort of cellular system or a system derived from cells with enzymes. These proteins can get all sorts of modifications done to them, specifically glycosylation, phosphorylation, other modifications. Different expression systems will make different variations, which can perform differently in the body. So it can be tricky to assay these things and show that your formulation is exactly, and your substance is exactly what this other biologic that's on the market is. For a small molecule, it's a lot easier. The analytical techniques, NMR, mass spec, other things like that, are very solid. For assaying large biologics, not so solid and much more difficult. So the FDA has indeed, though, got some biogeneric regulatory framework going, and a few of these are starting to be approved, but it's a slower process. And companies have jumped on this because they realize that having a biologic, like a good antibody drug, has for some years now effectively meant a huge patent extension.
Speaking of the FDA and new tracks being created for approvals, do you have any ideas of how the FDA could be improved?
Hmm. I have gotten a lot less bold about my big ideas over the years because a lot of people have been trying to think of big, bold improvements for things like this, and it's hard to come up with one that someone else hasn't tried or hasn't run the numbers on or doesn't have an example of it failing badly. One thing that the FDA has done more of in recent years is the accelerated approval process, especially for very severe or life-threatening conditions. They'll say, okay, you guys got some phase 2 data. It looks like this compound probably works. And that's about all you can say after phase 2 is it looks like it probably works. They now, for a lot of indications, will give you a conditional approval, an accelerated conditional approval based on this. But the idea is that you must continue to run confirmatory trials. And that approval is subject to being rescinded if those confirmatory trials don't bear out. They just took one down recently like that, and there have been several others. But there's been a problem in that the confirmatory trials, many companies have dragged their feet about running those. And the FDA, personally, I think really needs to crack the whip more. They seem to be doing that. They seem just recently to be telling companies, okay, we will grant you this conditional approval, but only if you are already enrolling patients now for the confirmatory trial, or only if it's already running. Until then, you can just wait. Your approval will be here when you can come back and show us you've got the trial going. I'm okay with that. I want hard data, and I want more of it, and I want it more quickly. And I think that the FDA is completely correct to be holding the whip hand on this stuff. So that's the main thing I can think of is let's just make it where companies have to do the right thing. And the incentive is to generate that data.
Some people argue that Big Pharma's profit margins are too high. And usually when people are making that type of argument, they'll show graphs of how much Pfizer spends on marketing versus R&D. What are your thoughts on the current allocation of money within pharmaceutical companies?
Yeah. Oh yeah, that is a constant one. One thing to remember is that the marketing budgets that you see often include a lot of other expenses dumped into that because that's often SG&A, selling, general, and administrative all together. And companies do not have to break that down more specifically into what exactly was spent on the marketing and selling, so they don't. So these figures are often larger than they actually were in real life. But at the same time, if you go back and look at the percent of sales or the percent of profits, either one, that are spent on marketing and spent on R&D, go to some other industries and have a look. The drug industry spends a lot on marketing, but it also spends a hell of a lot on R&D as a percentage. Companies that we think of as great big technological innovators like Apple spend nowhere near a percentage of their revenues on R&D that a typical large drug company does. Nowhere near. I mean, like factors of 3, 4, and 5-fold lower. So we actually spend a lot in both directions. And that's one of the other things that makes the industry so weird. And the selling expense is partly high because of the patent lifetimes. We know that every one of our drugs, except some of the antibodies, is under a time clock. And we have this amount of market exclusivity, and then we may weasel out of a little bit, but it's going to go away. People will say, oh, no, come on. These are big companies. It doesn't really go away. Well, Lipitor was the single biggest selling drug in the world. It went generic. So after that, what doesn't go generic? So yeah, we spend a lot on sales, but we spend a lot on R&D is my answer to that. No other industry quite does it that way because no other industry quite has to do it that way.
In terms of the marketing budget, and specifically direct to patient marketing. What are your thoughts on direct-to-patient advertising?
I wish it had never happened. I suspect there are other people in the industry who feel like that, and I suspect there are others who regard that as heresy. But I think that has done nothing but lower the reputation of the pharmaceutical industry, the ability to run direct-to-patient ads. I know, of course, why companies do it, because it obviously increases sales. I mean, no one's stupid around here. They're not just going to be throwing money away like that. But I really think that it has damaged the industry's public perception that way. There's nothing that can be done about it now. I don't see that ever being reversed. But my own druthers, I wish it hadn't happened.
What about direct-to-physician advertising?
Yeah, that I don't think you're going to be able to get rid of, in the same way that people always go on about how we have to get the money out of politics. That's like saying we have to get the blood out of the circulatory system. I mean, you're going to have to get word of your drug out to people. I mean, back in the late 70s, when Bruce Springsteen's album Darkness on the Edge of Town, his follow-up to Born to Run, came out, he apparently told the record label, why can't we just have it appear in the stores? No advance warning, no advertising, no nothing, just word of mouth. And the record label said, yeah, sure, Bruce, we'll get right on that for you. And, pushed him out of the room. You can't do your drugs like that either. You can't just drop a new drug and let word of it gradually spread out as if it were a Taylor Swift album or something. No, you have to get it out there, and you have to get it to the physicians, often specialists, who are going to be the ones prescribing. These are the people who are going to be writing for your drug. So one way or another, drug companies are always going to find a way to let them know, hey, we have something that we think you should be writing for your patients if you're not now, and maybe you should be writing for this instead for that other stuff. I don't see how you're ever going to get away from that.
Let's play a quick game that was shamelessly stolen from Tyler Cowen.
All right.
Overrated, underrated. So we're just going to throw out a topic, and you can tell us if you think it's overrated or underrated, and give a little explanation if you like.
Okay, so is this overrated or underrated in my own opinion, not what I think the world values?
Correct. In your own opinion.
Got it.
So the first one is remdesivir.
To the extent that anyone thinks it's a useful antiviral drug, it's overrated. I think remdesivir has just not shown enough efficacy in the real world. It's a good idea. It's a reasonable looking compound, but I have not seen the performance in actual clinical data that make you think that's really going to be that useful in practice. Now, if we had no other drugs, if we had no vaccines, if we had no Paxlovid, if we had nothing else, sure, I would say, give me the remdesivir. It's got a small chance of doing something, but it's better than nothing. But as it stands now, no, I just don't think it's a good use of anyone's time and money. Now, that said, maybe the next virus, it'll kick ass, but not for the ones we have now.
All right. Next one, sugar substitutes.
Oh, yeah. You've seen that recent paper that says that they can alter the gut microbiome. I've never been a fan of the things because I don't like the aftertaste of them. I would just as soon have real sugar and try to not have so damn much of it if I can manage that. So yeah, overall, I would have to regard sugar substitutes as overrated. I know there are people who pound down ridiculous amounts of Diet Coke and stuff like that, and I don't think that they're particularly toxic. So, you get all those crazy emails from people. There used to be actual physical mails, usually written in green ink and all around the margins of the paper, telling you how these things were causing autism and Alzheimer's and this and the cancer and this. I don't think any of that is true, but they may well be messing around with our gut microbiota in ways that we don't understand. So, overrated.
The 21st Century Cures Act.
For me, it's just about correctly rated. I'm about neutral on that. There may be, if people think that it's this really amazing sea change and everything, then I think they're overrating it. However, it is messing around a bit with pharmaceutical pipelines as people reevaluate just exactly how they're going to launch some of their compounds. I think some of those changes are probably bad. Some of them might not be bad, though. So I'm going to say, more or less neutral.
Psilocybin for the treatment of depression.
Right now, overrated just because of the hype. I think it actually looks like it can be useful, but people are, I think, a little too revved up about it for a couple of reasons. One, we really desperately need new modalities for treatment of depression, especially the most severe forms of it, which is what they're aiming psilocybin at. Very, very much need that. And we don't understand beans at a cellular physiological level of what depression is and what causes it. We really don't. It's a black box inside a sack. So there's a huge unmet medical need. So people are jumping on that, and I don't blame them. There's also a contrarian take of, see, you sat on these psychedelics for years, and the cure was right under our noses all along, you fools. So there's a lot of people who enjoy that sort of thing. So I think they're a little too revved up about it too. I think that these drugs actually do look like they can be useful, but I do not think that they are the cures for depression that we've been waiting for all along. Damn it.
The NHS in the UK.
I think that people in the UK overrate it. You remember back when they had the London Olympics and they had that big opening ceremony with all these, parade stuff going through and all sorts of things? Well, they even had a section that was all these people wheeling out these circling hospital beds with, dancing nurses and stuff, because the NHS was this great achievement of British civilization. I think the NHS is extremely well-intentioned, but I don't think it's a miracle. I think it provides fairly decent care to a lot of people, but I think a lot of Americans, as much as we enjoy bitching about our own medical system, and God knows there's some things to bitch about, I think a lot of Americans would not enjoy seeing the kind of NHS decisions that are made. Just to pick one related to drugs, the UK explicitly looks at cost benefit when approving drugs. The US does not. We look at benefit and we say, okay, we'll figure out the cost. We would really set off a bit of argument if we took an explicit cost benefit into our system.
Semaglutide and other GLP-1 agonists for the treatment of obesity.
Ah, yeah. Whoosh. Slightly overrated, again, just because of the hype. Obesity is one of those things that also people are desperate to have something that actually works and something new. Obesity is a graveyard of drug discovery, almost as bad as Alzheimer's. It is a graveyard of failed ideas and things that turned out to be toxic. There's a lot of stuff that never even made it to market and crashed and burned well before then. A lot of stuff. I worked on some of them. So the fact that these GLP-1 agonists actually do show the level of efficacy that they do in controlled trials is, to me, very impressive. At the same time, it's not going to be the magic pizza pill, which is what I think a lot of people want from an obesity therapy. The pill that you take, then you can eat all the pizza you want. Thermodynamically, and from just a conservation of mass standpoint, that's going to be hard to achieve. But I think they really are remarkable, but they are not the instant obesity cure.
All right, last one, the lab leak hypothesis.
Oh, God. This is a mess because I do not think the idea of a lab leak is crazy. And it's not incredibly unlikely. I mean, there is sloppy technique out there. At the same time, I think the weight of the evidence so far is against it. So I do not and did not rule it out, to core. But I think that the weight of evidence toward a zoonosis hypothesis is stronger. So right now I'm on team zoonosis, and I got there by sitting down and looking at the data. I did not get there through my politics or my deeply held religious beliefs or whatever. So the problem is you cannot come down on either side of that issue without making someone else hate you.
Now for a couple questions about your blog.
Sure.
How did you end up starting to write in the pipeline?
Yeah. Even going back into high school and before, I had always been very quick with written or presentation type assignments. It never really bothered me to get up and give a talk. Hell, I enjoyed it. It never really bothered me to have to write a paper. I'd sit down and write a paper. Most of the things I turned in were first drafts. They weren't so bad. So I got finally, through college and grad school and postdoc and got into industry. Well, those are not rate limiting steps in drug discovery. I can tell you, they make your life a little bit easier when it's time to give a talk or write up some sort of report on your work, but they're really not that much use. So I thought to myself, if I do have abilities in that line, I'm not getting much out of them. I should try to get something more valuable from them. And this was in the early 2000s, 2002. And I was starting to read some of the early blogs at the time. I thought, I'd like to do that. What should I write about? And as I said at the beginning of the podcast, people always thought that my job was really interesting when I told them about it. So I said, I'll write about that. I think it's interesting. There's a lot to talk about, and no one knows beans about it. Now, at the same time, I thought I'd run out of material in a year or two. So obviously I was wrong about that, but that's what got me started.
So now that you've written about pharmaceutical development for the last 20 years, on what topics were you the most prescient, and on which topics were you the most mistaken?
I can think of one that I was mistaken on right off the top of my head, when the idea of super enhancers in gene expression came out. I was skeptical that all these non-coding regions in the human genome were as important as they looked, because they could be varied up quite a bit without any particular change. So I was really going to be dogmatic, and I was dogmatic about it. I said, if you can mix up the sequence and nothing happens, then I don't believe these things are functional. That's incorrect. And part of it has to do with the lack of imagination on my part, in that there are even proteins like that. You would think that by rearranging the amino acids in a protein sequence, you would get a different protein. And God knows, most of the time you do, a worse one. But there are proteins that have long disordered regions on them where, and we've shown this by experiment, you can rearrange them as long as you keep roughly the same kinds of amino acids. It doesn't matter. They work just as well. So I was not really thinking in terms of the large scale organization of these systems. I was looking more down at the sequence level and thinking, this can't be right. So I was completely wrong about that. One where I was prescient was on approval of Alzheimer's antibodies. 10 years ago, I was complaining about Eli Lilly trying to get one called solanezumab approved. They ran like 3 phase 3 trials. I cannot imagine how much money they spent trying to push solanezumab across the finish line. And I kept worrying about this as, frankly, what an economist would call a moral hazard, an incentive to do the wrong thing. Because I thought, if they just push and push and push hard enough, they might get one of these trials finally to read out, maybe even by statistical accident, to show a tiny bit of efficacy. Then they'll run to the FDA and say, approve us. It's the first thing for Alzheimer's. And then if they do, millions of people will line up and throw all the money in their bank account at it because it's the only thing for Alzheimer's, even though it does nothing. I'm still worried about that. But this recent fiasco with Biogen and Eisai actually cheered me up some. The FDA, wrongly, in my view, approved it, but Medicare said, no, we don't actually believe this does any good. We're not going to pay for it unless it's part of a confirmatory trial. So I saw this coming, and we're going to see more of it.
What do you feel like makes Alzheimer's so difficult as a disease to treat?
Oh, man, that's a good question. I used to work in Alzheimer's. That was one of the first things I worked on in the industry. I started off with the tail end of a schizophrenia project, then I did Alzheimer's in the early 90s. And what I tell people is, if you had gone back to 1992 or so, 30 years and told me, beavering away in the lab, hey, guess what? I'm here from 2022 and we still are arguing about whether amyloid is the cause of Alzheimer's. Cool, huh? I wouldn't have taken that real well. I would have also done the math as 2022. Crap, I'm going to be 60 years old. You're telling me still don't know shit about Alzheimer's? Oh, God. So you wonder, why has it been so hard? And I think one of the big reasons is that we are the only animal. There is no animal model to do phenotypic screening on. We don't even know enough about the etiology of the disease to set up a real animal model. There are things about Alzheimer's we are missing. I think the final theory will have amyloid in it, but amyloid by itself cannot be the answer, or we would have had a success in the clinic by now. So I think that's part of the problem too, is that there has been a very plausible, very actionable hypothesis, amyloid, A beta, 1 to 42, and it has diverted the field for too long. It has just been too beautiful and too tempting. You just look at it and say, my God, that has to be the answer. But it's not. It just isn't. And we're thrown back on what our second choices are, and our second choices aren't very good. None of them are as convincing as amyloid, but amyloid's not it.
What are your thoughts on academic journals and how they've changed over time?
Yeah, there are too damn many of them. It has become, because of H indices and citation counts and stuff like that, it has become the academic currency for promotions and things like that. So it's a problem in that the prestigious journals know that they're prestigious. They know the power that they wield, and everyone else knows it too. So how are you to do anything but charge appropriately for that amazing power that you have? So we're in a nasty sort of vicious spiral here, where the journals can charge a lot of money and people feel like they have no choice but to pay it, because that's how they're going to actually advance their careers, and there doesn't seem to be any other way to do it. More people then start journals. Now, no, they're not going to start another Nature or Science or NEJM or whatever, but they'll start journals because everyone is trying to publish all the time. And that brings on a bunch of sleazy, totally dishonest stuff at the low end of the spectrum. The so-called open access journals. Now, I don't mean that open access is so-called. There are very good open access journals, but you have to pay to publish in them because they don't make their money by subscription. So there are page charges and article charges. Well, a bunch of real, con artists looked at that and said, really? Page charges, article charges, how delightful. Why don't we charge people to publish every single thing they send us and not spend any money at all evaluating it? And of course, there's a lot of that crap out there too. You can send random words into some of these people, and as long as your check clears, they will publish it. They have published it. People have scammed them over and over with this stuff. It's ridiculous, stupid, embarrassing, but it goes on. So scientific publishing is going to have to change. It can't go on the way it's been going on. It's unsustainable. No one is going to, a lot of people are not going to be happy about the changes, but I do not see how it can keep on the way it is now.
What biomedical technology are you most bullish on at this point in time?
Oh, yeah. I'm actually pretty happy about all this stuff, because right now, from a drug discovery standpoint, in my entire career since '89, there have never been so many really new modes of treatment being developed at more or less the same time. You have all these gene editing things like CRISPR. You have various kinds of cell therapies rapidly advancing. You have things like targeted protein degradation going into the clinic. That same principle behind TPD of enforced proximity is being used now preclinically to do all kinds of crazy stuff in cells, things we never dreamed of. We're rearranging the contents of the cell and bringing together proteins and enzymes that never would have seen each other to do things that they never would have done. So it's a great time to be doing this stuff. I'm really happy that my career has lasted long enough that I'm still doing it during this time. So I'm just kind of generally bullish. At the same time, not all these things are going to work. And definitely not all of them are going to work on the first try. My biggest worry is that something really nasty happens with one of these technologies, like happened in the early days of gene therapy. Jesse Gelsinger is the person who was killed, a patient who was killed by the early attempts at gene therapy. I always keep that name in mind. I worry that we're going to have another Jesse Gelsinger moment because you can't see that stuff coming. By definition, you can't see it coming. I hope not. I hope things go smoothly, but we're trying a lot of new stuff. Some of it's going to be spectacular and some of it is going to implode. It's not going to be dull.
Without a Jesse Gelsinger, do you develop tocilizumab as quickly?
Oh, yeah. That's a good question. You know, the poet Randall Jarrell had a line where he said, you can't make an omelet without breaking eggs, but that's what they tell the eggs. I don't really want to go there because I don't think that Gelsinger's death was a direct factor in speeding up some of this stuff. So I don't think there's a direct connection there. But yeah, without clinical failures, you don't get the clinical progress. Now, of course, here in Alzheimer's, we've had a failure after failure without much progress, but it's a little bit like, you know, if you're going to get something clean, you have to make something else dirty, but you can get everything dirty without making anything clean. For the most part, though, you have to have failures in order to show you what you're doing wrong and correct course.
And on that note, we have a few rapid fire questions to finish out this interview.
Fire.
So, number one, what important medical truth do very few of your colleagues agree with you on?
I don't know. I think my colleagues agree with all my extremely well-informed opinions. I'm not sure if I can come up with one where I am such a contrarian that almost no one agrees with me on. So yeah, pass.
What important scientific article do you believe more physicians should be familiar with?
Oh, that's a good one. I think that more of them should read a very irritating article called Can a Biologist Fix the Radio? And it's a sort of a diatribe against reductionism. And it talks about how if you were a biologist fixing a radio, you would probably just rip all the parts out of it and assign them to different weird categories without any understanding of electricity or resistance or voltage or any of the things that actually make a radio run.
If you had unlimited funds for a single randomized controlled trial, what would you fund?
Oh, boy. That's a tough one. I honestly would be tempted to try it on something on generalized diseases of aging. I know the people who are looking at metformin for that have been trying to get funding to put together a big metformin trial to see if it really does have effect on generalized aging and cellular senescence. I might turn around and fund that. Who knows?
What would you be doing right now if you hadn't become a chemist and a blogger?
Oh, yeah. I wondered that too. Because, I mean, it all looks inevitable in hindsight, but none of it was. Hell, when I got to college, I didn't even know I was going to major in chemistry. I wasn't 100% sure I was going to major in science. I don't know what I'd be doing. I might be an unsuccessful novelist.
What would you be writing about? Spy novels?
God, who knows? I'd probably be writing all this weird crap that no one would want to publish and no one would want to pay for. I'd be writing all this convoluted stuff like Borges and Nabokov are trying to do that and failing.
On that note, thank you, Derek, for joining us on the External Medicine podcast. Before we close, can you tell people where they should go on the internet to find out more about you?
Yeah. Well, most working days, not today, I had too many meetings, but most working days, there are new posts every day on in the pipeline, and that's at science.org derek.lowe.com/blogs/pipeline. But if you just Google Derek Lowe and in the pipeline, it'll come up fast. The only other Derek Lowe you'll get is a guy who used to pitch for the Red Sox. And I could turn him into a chemist before you could turn me into a mid-inning reliever.
Thank you once again. We really appreciate you coming on.
Oh yeah, thank you so much.
I enjoyed it. As you can tell, I like talking about this stuff.